to the Weekly Bioanalysis, a KCast Bio podcast.
Hello, and welcome to the 99th episode of the Weekly Bioanalysis, the official podcast of KCast Bio. KCast Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industry for over 45 years. My name is Dominic Marino, and I've been in the LBA and biomarker space for around 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for around 30 years.
John and I are members of the growing scientific services team here at KCast Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCast Bio's services. We are amped up to have you listening to the 99th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus of a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun podcast. To kick us off, for podcast number 99, John will go over today's podcast topics a bit before we jump into the news and resources. John.
Yeah, hi, Dominic. As usual, we'll be going through some news items, trying to look at where medicine is heading. And then our main topic today is breaking silos, driving project execution through continuity amongst bioanalytical teams. As we've matured as a bioanalytical organization, as we're dealing with entities that involve a lot of overlap between the teams, we've worked to build a consistent approach between the teams and use that to help provide a better product for our customer.
John, exciting topic to today, but let's first jump into our news and notes. What's the first topic, John?
So first thing is just to break things in easily. Every time I put an agenda together, I don't want to talk about GLP-1s again, but then there's always so much news about them. And this is just there's a group been looking at how GLP-1s help the heart relax. So it's not only a GLP-1s there for diabetes and weight loss, but they're also showing that, particularly in data from rats and mice, that GLP-1s can support heart attack recovery. And this is all down to helping blood vessels relax. So basically the mechanism that they talk about for heart attacks is coronary arteries filled with plaque become clogged and cause a heart attack. After plaques purge from the system, blood vessels can remain squeezed too tight in a state known as no reflow, causing further complications even after the immediate emergency is passed. And so the group at University College London basically gave Exendin 4, which is a GLP-1 mimic, to rodents with constricted blood vessels, and showed that this actually helped the capillaries relax back to their normal state. And it's basically showing, it's digging into the mechanism, and as yet another example where GLP-1s might help, even though it was something that they were never designed or never really thought they'd be helping here. But the more we learn about them, the more effective they seem to be in various parts of the body.
Yeah, it's fascinating, John. You would figure losing weight in and of itself is going to help you with heart disease, but now this is saying GLP-1s have direct mechanism of action.
Sure, exactly. Yes,
and if you go back to even a lot of the heart medications, which eventually went on to become all sorts of the Viagras and the things like that.
Yep.
This to me it's not. It is surprising, but not surprising. If that made any sense at all. I mean, I know, like I said, I think there's clear evidence that if you lose 15, 20% of your body weight, or really appears to be a miracle drug for those that are really obese. But this finding is pretty fascinating, John. The way it's kind of helping. I guess it's a vasodilator, right? Or is it?
Yeah, I think. I mean, they sort of talk about how it links the blood, the brain, brain, the gut, and the heart together in terms of the mechanism. So they were looking to understand why blocked blood flow, like limbs, can prevent heart attacks later on, which is a thing known as remote ischemic precondition. I'm reading this. This is all new to me. And they determined that when the brain detects blocked blood flow, it triggers the release of GLP-1 molecules from the gut, which then bind to receptors on the surface of pericytes that are part of the blood vessel architecture. And by activating the potassium transport mechanism, the GLP-1s induce the pericytes to relax. So it's showing how interconnected the parts of the body are, and just helps the understanding of the role, their role in this.
It's like it's all part of that axis in the body that goes from like brain,
sure, yes,
all through the body, and to me, it's a really incredible finding. But it's also like, growing up, my mom used to always say, hey, let your stomach settle, let the food in your stomach kind of digest a little bit, and you won't be so hungry, right? And I think this just further proves that it does. That's why you overeat, because you don't realize you're full, and if you just slow down eating, it'll kind of do it. But this kind of, I think the GLP-1s have many ways of benefiting, and this is just yet another one where it really does help your body understand that you're full. It's interesting because if I'm reading this right, it was semaglutide and tripazitide were already approved for cardio risk, right? So
they have been, absolutely, yes.
They're expanding it. So
I think this is just more looking at the mechanism behind it. So I think that's what because like you say, the GLP-1s pop up a lot, and at least just to get ready for this, there was one about talking about, and we've touched on this in the past about the potential in terms of helping with drug and alcohol abuse and what's going on there. And there was at least one big study that was looking at data of people taking GLP-1s versus other diabetes medications, and showing that the rate of alcohol and drug abuse really declined, and this was just going through historic data relative to the other diabetes medication. So there's going to be ongoing studies looking into, can GLP-1s help there. And then I also got sent a link, which I haven't done any digging on, but it's also because of the weight loss associated with GLP-1s, it puts less pressure on limbs like your knees, etc. So helps in cases like arthritis. So it's like an indirect effect, but it's a genuine effect. So it's like, if I will dig into that a bit more, see, because it actually directly affects me. But it's that whole thing of you've got a benefit, but there's obviously downstream benefits. Some are directly related to the mechanism of the actual drugs, and others are related to the effects of the drug. And like I say, you lose weight, it's less pressure on joints, etc. So there's a lot of interesting stuff just popping out at the moment in terms of these medications.
Yeah, John, just fascinating how effective these GLP-1s are. This article talks a little bit about potassium and how that's the mechanism of action, but it's really interesting that anytime you've got blocked blood flow, which is ischemic, right? That is a really bad thing. And you talked about how you're kind of dealing with some of that yourself, but that's part of the reason why I sit crisscross and do all sorts of crazy things to help make sure I've got good blood flow. But GLP-1s are they really do appear to be some miracle drug for a whole host of sort of diseases. And who knows, maybe it'll be in the water like fluoride over time, right? So anyhow,
who knows? I'm not going there yet. Let's move on.
Yeah.
So the next article is, study suggests long non-coding RNA has potential as a new class of genetic medicine. So scientists are now developing synthetic versions of a historically overlooked molecule that could unlock a new class of drugs. These are basically long non-coding RNAs that control how genes are regulated. They're more abundant than the typical protein coding genes, but there's been very little research on them. And so this is work out of the University of Toronto, is shown that three long non-coding RNAs previously known to control immune system genes can reduce inflammation when administered to human cells and in mice. So again, it's early work, but it's interesting because this actually goes into they've shown effectiveness, but the actual long non-coding RNAs actually have not a very long half-life, so it's how do you then make them effective. They could increase the half-life by capping it at the two ends of the RNA, but that then that eliminates the effectiveness. So they've actually now in the process of replacing within this, or adding and changing things within the sequence to maintain effectiveness and increase the half-life. So I'll pass it to you, and because this is obviously much more in your realm.
Yeah, it's really these non-coding RNAs have always been a little bit of a just. If I go back to when I learned about RNA and DNA, they just said, "Oh yeah, don't worry about all that stuff. It's all just nonsense." And now we know that's not true. I think that maybe 10 or 15 years ago, or if you go back to like maybe 25 years ago when they first started doing the whole human genome, they started to discover that these non-coding RNAs had some potential to them. And really it's because it's involved in how things are folded. And, as we've fast forward to today, this is a breakthrough, John, for sure. I know it's at the basic level. It's really only, they've identified 3 of these non-coding RNAs, and hats off to the University of Toronto and Omar Khan, who long ago thought they must have some information to them. It turns out they do. It's really fascinating that, these synthetic versions of them, you kind of talked about how they can cap it. They'll figure that out, right? First,
absolutely. That to me is less of a challenge.
Exactly. It's amazing how I think of it as engineering, but just, it's no different than starting to pegylate things, and they'll find a way. But this is certainly seems to me to be a real future of, untapped, right? This is the next, if we think of it as like the, space is the next frontier. If we got CAR Ts, and you've got now, the in vivo CAR Ts, if you know what I mean there, with like AAV based CAR Ts or any sort of viral based CAR T, this seems to be the next frontier of medicine, John, right? These RNA, non-coding RNAs, and it's something that never sat well with me when I first learned about it. I'm like, well, why is it there then, right? And so
exactly.
On here, he spent decades researching it, and it's just a wonderful publication, John. It's still early, but I think it is the future of. I'd be surprised if in another 20 years this isn't going to be the breakthrough, or hopefully less, but it'll be a breakthrough kind of drug type.
Yeah, it's sort of interesting when it talks that, there hasn't been really much effort on these long non-coding RNAs, and it's been more trying to use small molecules to trigger an effect in them rather than to actually look at can they be used to do something themselves. And it's all about, we talked about the chemical modification, but it's more about delivery of in lipid nanoparticles. So then you're keeping your anti-inflammatory to keep basically any attack on them down, and actually have them do what they need to do. So to me this was like a really interesting sort of. It's something I've not really thought about because I'm on the small molecule end of things, but to have something where they talk about there's 20,000 protein coding genes, but scientists have identified roughly 36,000 long non-coding RNAs within the portion of what is called genomic dark matter within the genome. So there could be lots of potential here.
Yeah, one of the other things the article notes is like this is another sort of benefit of some of the COVID vaccine stuff, right? Mm-hmm. This is kind of coming out of that. All the, kind of focus that we had now almost 6 years ago, right? That's become, it's just another benefit of all that research collaboration that we had talked about years ago. And this is just another case where the stabilization of it is some of the tricks that they learned from mRNA vaccines and COVID vaccines, right? So really great stuff. I mean, this is, it's kind of got me excited, John. This is the future. And so, I talk to a lot of young up and coming scientists, and they often ask, where do you think medicine's going? And this would definitely be on my list of where I would focus on. Right? Yes.
It's very difficult to say it's like it's going in a particular direction because there's so many exciting new and different directions. It's like and even when you got all this new stuff, you still got your traditional small molecules that we've been living with for decades are still contributing to a very large extent. I mean, as we talked about, drug approvals last year were the great biggest proportion of new approved drugs were all still small molecules, so they have a huge part to play too. So we got all this exciting stuff, but it's also lots of developments on the traditional side of things. I mean, there was one article that I didn't obviously have not covering about the small molecules are sort of being resurgent in the rare disease environment. So like I say, it's although there's this kind of stuff could be could potential in the future, small molecules are still sort of carrying this there as well.
Oh yeah, no, the small molecules are not going away, John. We know
No,
that.
absolutely, yeah.
We've heard that. I can't recall a year where it ever wasn't dominated by small molecules. I think the lowest percentage was in the 50s, right? It was still always the largest percentage. Even last year, I think it creeped up to almost 70%.
Yeah, exactly.
Which is somewhat surprising, but it is a you wonder if it's just tackling it with. In the future, will there be like some sort of non-coding RNA plus a small molecule or something like that, right?
Sure. Yeah.
Fascinating stuff. But
Let's
what do
move
we
on.
got? Yeah, I think we got one more.
Yeah. Well, we got large-scale gene editing tool could power next-gen genetic meds, and this is a new method for safely inserting large chunks of DNA into genomes. And again, this has been done in mice, so again, it's early stage, but it's potentially paving the way for next generation of gene editing medicines. So this group. Let's see. The technique was pioneered by a team led by Connor Tao and Benjamin Kleinstiver at Mass General Brigham. And they're using circular single-stranded DNA rather than typical double-stranded DNA, and they call it install, which means integration through nucleus-synthesized template addition of large lengths. And so it basically, this, we'll call it install because we're not going to talk about it as integration through nucleus-synthesized template addition of large lengths very often. And it's capable of inserting DNA into genomes of mice and lab-grown human cells. And then they're actually looking to see what they can do in terms of new medicines for liver diseases, blood disorders, and metabolic conditions. And I think, again, this is... the key issue install addresses is the immune response to large chunks of double-stranded DNA, which aren't normally found floating freely inside the cells. DNA in the cytoplasm of the cell usually means something is wrong, and so when a cell detects DNA outside of its proper home of the nucleus, the cell launches an immune response that causes downstream toxicity. And so, like I said, this approach takes the single strand. And this was, again, I love the way it always comes back to nature. Tao and Kleinstiver were inspired by certain bacteria and viruses that can naturally insert circular single-stranded DNA into double-stranded genomes. And what this means is, as you read more into it, it's actually, it's sort of, these are like large edits for parts of the DNA that are wrong. So you could actually take out rather than doing like a single site or whatever, it actually can replace large chunks of the DNA and can possibly address more conditions with that edit than just your single site change. But I'll pass it to you because
it's amazing, right? So there's so much to kind of unpack. Again, this is another kind of based off the gene editing, CRISPR, the next
exactly.
Next iterations of those, and you really touched on the beauty of really understanding nature and how, hey, when a cell has circulate, if you've got double-stranded DNA outside of the nucleus, that cell goes into like a death mode because something's really wrong. So if you try to treat with that, you by default, maybe maybe even part of the mechanism of action might kill the cell, right? So very cleverly, they came up with this single-stranded version of it and packed it into a particle, called it install, and it's still some early stages. I think the company's Verve, if I read that right, who's kind of working on this. And
I think the nerve is actually the nerve is actually it's a CEO of Nerve talking about the technology.
Okay.
So it's the guy called Musunuro. I think he's the CEO of Nerve. I've now lost my.
Yeah, it's Verve, but I understand what you're saying. So
Yeah,
it's not them as a company; it's still in the real early academic stages.
Yep.
But it just goes back to like, man, this is going to be great. It's probably still a fair amount of time away, but one of the really take home messages is they talk about, instead of needing 100 tools to treat 100 mutations, which, by the way, oftentimes, if you've really studied cancers and tumors and things like that, it's rarely is it a single kind of mechanism. It's often hundreds. That's why you survive, right? It's part of our our survival system as a species, is to be able to have so many redundancies that when you do start to have mutations, you don't really know until there's just so many. And so they talk about how instead of needing 100 tools to treat 100 mutations, it's one tool to treat those 100 mutations. And the science behind it is still a little elusive to me, but I think, as you said, it's just they just replace large gaps of mutated sections and they're able to repair it. And it's just still, to do it systemically is still somewhat hard to envision, but it's really, really cool nonetheless.
Absolutely.
Yeah. But John, my mistake. It seems like you got one. I don't know if you have anything to add. I don't.
I have nothing else to add. I thought I love on this section where we can talk about things in the future, and at some point it will impact us or someone within KCAS bioanalytically, when as more of these medicines come in. This last one's a bit of a throwaway one, but it's still interesting. This is I love it because it often pops up in fierce biotech and endpoints, different approaches that people are taking to marketing, and quite often it's finding someone like a prominent actor or a sports person who have a certain condition, and so they want to help be a spokesperson for that condition, which obviously is great because it humanizes it for people who don't necessarily know about it, or it gives them someone they can relate to. But this is an initiative in Japan. It's between Eisai, who I don't think we've ever spoken about in the podcast before, and the headline is Eisai taps Pokemon app for healthy sleep awareness collaboration. I'll say that again. The headline is Eisai taps Pokemon app for healthy sleep awareness collaboration, and I can guarantee we've never talked about Pokemon in this podcast before. But basically, so Eisai and Pokemon have got together, and they're trying to inspire or help people develop healthy sleep habits. As according to the article, according to a new team up between Eisai and Pokemon, when it comes to healthy sleep habits, people of all ages have got to catch them all. So this is happening in Japan. Basically the Eisai is partnering Pokemon Sleep to basically push on the awareness of the importance of good sleep. And in Japan, they're saying that 12% of adults experience insomnia and about 40% of people sleep less than 6 hours per night on average. So what they're trying to do, they have a mobile app which is placed on a user's bed overnight to track sleep. It uses the nightly results to not only provide sleep quality reports, but also to generate digital rewards, unlocking new characters and using those connections to nourish the famously sleepy character Snorlax, who serves as the app's mascot. And so, to start things off, Eisai will distribute a picture book called Snorlax's Dream to Japanese medical institutions that offer sleep disorder consultations. And they're obviously pushing Eisai's expertise in this area. So this book takes place in the world of Pokemon Sleep, with Pokemon characters discussing the importance of and tips for healthy sleep while they embark on a quest to find Snorlax. I just loved it. Like, it was so out there, and it's that, and people will relate to it. Having visited Tokyo and seen, you've seen like Pokemon raids, etc. People of all ages just are it's a big deal there. So I think this is very, very interesting.
So if I understand this right, it's like a game. The more you sleep, the more you benefit in this Pokemon kind of app thing,
I
right,
think
John?
so. Yeah,
Yeah.
yeah,
So it's really it's not a there's no like medicine involved here. It's just more like a psychological kind of approach towards it. I think it'll work, but those are some staggering numbers in Japan in terms of 12% of adults have
sure,
insomnia. That
exactly.
seems
No, I
high.
know that's a
And
large group of people.
40% with less than 6 hours per night. I don't. I wonder what it is in the U.S., right? But these types of games could have profound effect on someone's ability to sleep, right? Because then they just want to get more points on the game, and I don't know. It's really it made
Although
me just go ahead. Sorry.
sorry, I think the positive results are related to you having a good sleep. So I think it's like
Yeah.
if probably if you've got a better night's sleep, you've got more chance of generating the digital rewards. I don't
Right.
think you won't get anything if you're awake all night watching to see what happens. It really is related to your sleep quality, etc. I assume. I
Yeah,
like
no, I think you're right. I think you're right, and I think that in part it might just relax you a little bit or give you some incentive to sleep better, and those types of things can have profound effect on efficacy. So it's really it just made me laugh when I looked at it. I thought, what a clever way to do it. And as you kind of alluded to earlier, usually it's some sort of actor or sports thing. It's really I think this could really be very beneficial. I've never. I mean, I was born in Japan, but I haven't been there since I can remember. But if it's a huge thing like you kind of describe it, I imagine Pokemon's enormous there. It really could benefit these, approximately, I don't know, about 40%. I'm sure the insomnia people are in the 40% that are less than 6 hours sleep, but that's a huge population that could
yeah,
have some
no,
benefit
absolutely.
just by using a little app to kind of settle you down. It's just it made me laugh, and I thought it was a great story, John. So anyhow,
Yep,
yeah, John, maybe we'll move into our main topic today. And
sure.
to me, it's near and dear, right? It's a pretty exciting one. We're gonna talk about breaking silos, right?
Yep.
Driving projects through execution through continuity amongst bioanalytical teams. So, John, we got a jam-packed agenda here, but it really is like I really the first topic is doing things the old way. Different approaches to different technologies, and I don't know if you want to get started first, but I
I
certainly have
can,
my thoughts on ligand binding assay for
yeah,
sure.
I mean, to me, it's definitely the older model has been, mass spec and ligand. I'm going to particularly focus on mass spec and ligand binding simply because there's the overlap there. They've each had their applications, and I mean, it's certainly true of the company I used to work for. There was very little overlap between the two teams, and I mean, they were geographically separated, which didn't help. But because there was little overlap in terms of clients, in terms of kind of entities we were working on, the practices between the two groups were night and day. We did not talk to each other about what was best for documentation, what was best for in terms of reagent tracking or anything like that. It was very much, we do things our way and the other team does things their way. And ultimately what we're seeing is, the more we go into these more complex therapeutic entities, it breaks down the walls. And it's not just the therapeutic entities, as we'll touch on, it's also like sampling and just dealing with all the complications of these more difficult to deal with um, dealing with these more difficult kind of analytical challenges. And then, of course, you got hybrid LCMS with an LBA have a lot of rooted technologies where one can benefit from the knowledge of the other, etc. So that's the way I wanted to start it, and we can take it from there.
Yeah, no, it's so you kind of touched on it, but if you go back to like 2010 range, or maybe even a
Mm-hmm.
little earlier, biologics are really starting to
Sure.
hit the market, and there's uh, any sort of guidances or ways to validate methods are predominantly mass spec geared.
Yep.
It took until like 2018 to harmonize that. It took like a decade, right? Remember the 2018 bioanalytical method validation guidance from the FDA sort of tried to combine the two, and there's still some kind of nuances or differences between a traditional mass spec approach, a hybrid mass spec approach, which is adopting LBA, right? The
Sure.
criteria and things like that are kind of 20 to 25% variability on the in terms of acceptance criteria. But then now comes PCR, and now we're really struggling with that to try to get that into it, because it's still trying to. At the time when we were looking at biologics and small molecules, it was like trying to fit a square peg into a round hole, and it took a long time to kind of create some common ground. And now PCR hits the landscape, and it's definitely not applicable. Although we're trying, there's some movement. There's been some AAPS papers and things like that, and KCAS Bio's on that. But there's still some things in there that make it hard for the three fields to kind of really have this unified approach. And it's, John, I don't know if you knew this, but PCR doesn't really do multiple replicates of the same sample. And what I mean by that is, in a ligand binding assay, if you say you do duplicate replicates, that's the same, kind of diluted sample or prep sample plated 2 times.
Sure.
Well, in PCR, that they never do that, because the platform doesn't need it. When they say duplicate replicates, it's 2 separate preps. So even that kind of stuff is tripping up just kind of trying to talk about validating it. And, as a group KCAS Bio provides a service. So when we're talking to clients in the PCR space, we have to be careful when they say they want multiple replicates, because it has a different meaning.
Sure. Yeah, and that would be the same
example. Yeah.
That would be the same for LCMS. I mean, if we're doing 6 replicates, for example, of a QC in a validation run, that's 6 separate extractions. So they are 6 different samples, and they would be taken from different tubes. So it's or 6 separate aliquots to begin with. So again, it's they are not the same sample. They're treated as different throughout the course of the run.
So in LBA, they'll do 3 preps of a sample and plate it plate all of them in duplicate replicates, all 3 levels of QC. So it takes up a lot of the plate for an A&P batch.
Yeah.
But it's just those subtle things that we just got to be careful when we're, kind of getting into the weeds on what exactly you're doing in a validation or a qualification. But and I don't know. To me, when I thought about even doing things the old way, different approaches to different technologies, I even thought of, like, within the LBA space, this desire to get ultra sensitive biomarker and even PK now, right? The ELISA platform is evolving to becoming potentially a very powerful PK tool for this big push to reduce the number of animals. Well, if you can really use a much smaller sample size to get incredibly sensitive PK with a large dynamic range, that solves a lot of problems. So that's one evolution. And then MSD came out with a booster pack, which is fascinating. And that's to take a standard Vplex or Splex or Rplex, whatever it might be. And this booster pack is putatively supposed to get you about anywhere from 5 to 10x kind of improved sensitivity. So really cool stuff there. But I don't know, John, that was, when I read that, I kind of thought, that would be a good thing to throw in there. But, I don't know if we have anything more about that. I don't know where you want to take us, but certainly more than willing to.
I mean, to break off from the recording, I think this is just about where we were and where we going, because there's much more and more overlap the more we work on projects. So we could chop this piece, but I think that to me is what this is about. I know this was all driven from marketing, but that's the best way I could interpret it. So we can say what we've input. That's why I threw in the scientific advisors, because to me, and I will pick this back up within the actual recording, to me, because we're there in calls together, we're hearing the same thing from the clients, but we're immediately thinking how we can work between teams for a success of a project.
Good deal.
Okay. So that's where I'm coming. That to me is what this is about. It's always more difficult when someone gives you a topic to talk about than when you're actually setting it yourself.
All right, we can pick back up then.
Yeah, so let's move on with the agenda. So for me, the way we're, like, say, our in recent years, the way the industry's going, in terms of, the entities we're working with, it has made the teams work more together. It's facilitated by the fact that we're all in one building, and so it's easy for us to talk to each other. But ultimately, before that, even before the project starts, we because of the team that we're part of, the scientific advisory team, we're there in scoping calls. So if it's a traditional small molecule, it'd be me or Dawn, depending on where the project might be going. But if there's biomarkers involved, then we'll have LBA usually be involved. But as we get into things like ADCs, where there's likely to be multiple analyses involved or technologies involved, then we've got scientific advisors representing each of those service lines, but we're there. That's almost we're unifying the approach to the customer, and we're going to be taking the same messages from that scoping call, and basically that then we're already starting on the footing where the groups are thinking about how do we work this, and where the overlap is, how do we make that work as smoothly as possible.
Yeah, John, I think it's really part of the secret sauce here at KCAS Bio. And what gives us a lot of flexibility and nimbleness is having a scientific advisory team, which is dedicated to kind of servicing the commercial team. And so you touch on a really good point when a client might be a traditional small molecule, but there might be some cytokines that are needed. So someone like myself or Lindsay would join you on those calls. And then obviously complex drugs like ADCs are going to require both a ligand binding assay and a mass spec approach. And, that we kind of learn through osmosis or diffusion, right? We kind of,
Sure.
as we all are able to see each other present on a high degree of frequency, and it does allow us to kind of understand the regulations, right? So that improves our quality to understand some of the differences between, say, small molecule, large molecule, PCR, flow-based, all the kind of platforms and services that we offered. And it just, again, it really does allow us to kind of think about the service line we're in and how it can be harmonized across other service lines. It's part of the reason why we're prominent at WRIB. We're going to really have an active team at the GCC this year. We're leading a couple groups there, but that I think it is, when we topic of breaking silos, it is getting mass spec people to think, or at least realize, that LBA is over there and vice versa, right?
I agree, absolutely. Yeah. I think I remember my old. I'm not slated to my old company at all, but it was always an interesting experience for me when this happened. We were strictly a small molecule LCMS lab, and we were offered the opportunity to set up LBA with one of our established customers, who sort of said, we recommend this person. If you take them on board, then there's a good chance we'll send LBA work to you. But I think because we had such a small molecule LCMS mentality, we couldn't figure out how that worked, or how that person and a potential team built around that person would work within our environment. And in the end, we sort of walked away from the opportunity. And that to me that was 20 years ago, probably at least 20 years ago. And now you look at where we are, where it is really important that not only do you have both teams, but both teams are actively talking to each other and working with each other. It's as we've seen with science over the last 40, 50 years, it's moved incredibly quickly. And so it's things like it's just a reflection of that. The science has moved quickly, bioanalyticals had to move quickly, and the way we worked has had to change to accommodate that too.
Yeah, no. So when I joined KCAS Bio 13 years ago, John, what you described is exactly what was happening. They were heavily focused on mass spec. I remember when I first joined, some of the mass spec people were like, you should be quiet, LBA is not that relevant. Although at the time we were a 3-person team, and today we have 70. I'm very proud of the fact that team has grown so much in my tenure here. But that was really probably the first challenge I had, was just breaking that silo of trying to bring us all together and understanding that the the way drugs are evolving, it's going to be a combination of mass spec and LBA, and that's really been driven home today in the ADC space as well as even the peptide space, right? You have an immunogenicity
Sure.
component there,
Yep.
and so it really is something that our scientific services team allows us to provide good quality service lines that are able to bridge the gap between them, and it is a fascinating story of how the mass spec people, you don't hear as much of it today as you once did, say 5 or even 10 years ago, about how different they are. The community overall has done a good job of kind of understanding that you can have one guideline that can fit both of those platforms. But as I said, PCR hasn't quite gotten into that mix, and flow is still kind of out there just because of the very nature of the number of reportables. And now as the flow industry's grown, there's, you can have anywhere from 30 to 60 reportables with spectral flow or even higher. So it gets even more challenging where, I know we have some multiplexing, but that reportable piece becomes quite challenging in terms of how to manage the criteria of each. If you got, I don't know, even in 10 plex on an MSD or some sort of high 25, 30 plexes on Luminex, if one of them falls out, do you have to repeat the whole thing? And that's, that becomes real challenging from both a costing perspective and why would you repeat the other
Sure, absolutely.
analytes if
Yep.
they pass? And there's
Yep.
ways to manage that, and you need a good a priori criteria to manage that. So anyway, John, yeah, great. Why don't we can move along
Yeah,
a little bit in our agenda, because the next topic is really near and dear to me.
which one?
The handoff between
Oh, yeah,
method
sure.
development and validation. That is a really, that's something that I think is what we excel at, but it is something that is, on the surface it seems so easy, but it's
Yeah.
often one of the big choking points of bioanalytical development and validation, that the handoff between the two has to be smooth, or you end up in an investigation. And we've over the years done several things to help avoid that. And I would say today, I hope I don't jinx us, knock on wood, our investigations are at an all-time low because of some of the processes we've put in
Yeah,
place.
and I think I'll step back historically on that. I think there's been a tendency amongst pretty much all CROs, as they've got bigger, as you start off small, and when you start small, everyone does. Anyone, it was certainly true of us, my previous place. When we started, you did everything. So I think the only task we didn't do was log in samples, but we basically did the method development. We did the validation. We extracted samples during sample analysis, and we dealt with customers, etc., etc. But once you become above a certain size, that becomes unmanageable. So we, in common with virtually every lab I've spoken to, and it's also recognition of different scientists of different skill sets. Method development scientists think a certain way, and I'm not. And then people who are more in the production, i.e., validation, sample analysis, they're more driven by other things. I mean, my driver was how quickly could I crank through a bunch of samples and move on to the next thing, but still, give good quality data and that complete package to the customer. The method development didn't interest me quite the same way. So, long story cut short, the model that most labs have gone with is to have a method development team who are focused on the early piece, who then hand off to the validation team. I know when we made the split, we talked to one customer who really didn't want us to do that. Because it was because the key point is that handoff from method development to validation is where things could go very wrong. And if you don't do it right, it ends up a finger pointing exercise between the two teams when something doesn't work. And we definitely successfully managed that with my old team. But what I like, why I put this in as an agenda item, at least within the LCMS team, the handoff is quite thorough in terms of there's side-by-side working between the development person and the validation scientist to ensure that the assay is working within the validation scientist's hands before they're before that moves forward. Plus, the development scientists are very much involved in terms of being available in case there are issues. So it really is it isn't just a get to the point, the development's done, they hand it off, and you never hear from the method development scientist again. It's there's still involvement such that when if you do see issues, which you generally do with analytical chemistry, there's a support network there from the scientists who originated the work.
Yeah, the industry, and obviously we have that pre-validation step is now critical.
I meant to touch on pre-validation. Absolutely, that is a huge part of it all.
Yeah, it's almost if you look at the 2018 guideline, it is almost a mandatory step that needs to be done. If you haven't done a mini validation in development, even those batches can be reviewed. That becomes a real critical piece to that smooth handoff. And I know we've been here so long, we've gone through different iterations as we've grown from having a standalone development team that did the handoff and they were intimately involved, and that's kind of how I think we do it in mass spec today.
That is, yes.
Yeah, but now in our LBA team, that we kind of reverted. We moved away from that about 5-6 years ago, I think, to having where the method development scientists, because of the nature of LBA, sometimes it's incredibly tricky. And what I mean by that is, for whatever reason, the development scientist might it might not always be an analyst 1 or 2 can pick that method up. So a development scientist can be part of the validation in our LBA team. We try to avoid that, but it is an option. So it gives us a lot of flexibility on the ligand binding assay side, but it doesn't happen that often these days where a development scientist has to be part of the validation, but it is an option that we can do.
Sure. Yeah.
And for the most part, and they really pick it up the moment the method's developed and they start doing the pre-validation. They do introduce the analysts are part of that pre-validation. So it's a real nice smooth handoff. And I can't think of any investigations over the last few years in ligand binding assay that were due to some sort of variation between analysts.
Sure, absolutely. Yeah.
It's something that I think we're fortunate that we've worked at KCAS Bio. We've invested so much into it. I know other CROs probably are, but we've really found a nice groove there to ensure that we have a smooth handoff, and it's something that, again, we're proud of and really don't have a lot of issues with it, because it creates a lot of time. Time is of the essence. Any delays in validation can be critical towards delaying submissions. We avoid that at all costs. We're very good about avoiding that. I feel like we kind of beat this to a pulp, John. If you have anything else to add there.
Well, we can move on. Next piece we were saying about, and I've alluded to it plenty of times already here, is about new entities that we're dealing with, and this comes to assays or analytes like ADCs, where you've got the large molecule portion and then the payload being small molecule, which is almost automatically done by LCMS, but then the large be the ADC and the antibody can be done by either LBA or hybrid, and there's no unified approach because we talk to some customers of a preference for mass spec, others really want to stick with the LBA. But as I was thinking about this, ultimately it's these kind of molecules that drive that need for consistency and continuity between groups, because if one group can't execute at the level of the other, it does then cause a problem in the client's eyes, and that might mean that long term you're not working with them because one group let the other down. So it becomes really important. Sorry, excuse me. Becomes really important that both groups are delivering to the same level. Although it's part of our drive within the company to standardize between groups to a large extent. You can't totally there is obviously some difference in the technologies, etc. I mean, you can't completely standardize, but if at least you're consistent to a large extent between the groups, then that presents that good unified approach to the client. But you still got the execution piece, and if one group can't do what the other's doing, then that causes an issue. So it is really important to keep an eye on that. And like I say, that in that case, it's the therapeutic is driving it as almost as much as we're trying to drive that consistent approach.
Yeah, no, you spot on. I mean, things like ADCs, ARCs, any sort of conjugate technologies, Frankenstein-like molecules that where you might have something that makes sense to measure a portion of it by mass spec, a different portion by LBA, or even obviously the immunogenicity is going to be done by LBA.
Sure.
That is that kind of what you're alluding to is, even if you're a mass spec expert or an LBA expert, what we want to provide as a service is to make that seamless, so it doesn't feel like you're working with two companies. And that's something that really I think separates us is our ability to make it a little more seamless around, so that when one group gets on the phone, they're presenting and showing data the same way as the other group, John, right? That's kind of what we're talking to about how to just make it a more, because bioanalysis is it's a combat sport. You've probably heard me say that a number of times now. And the last thing you need is, kind of trying to be the client, trying to put one hat on for one group and one hat on for another group. We've worked really hard to make sure that it's a similar experience. And some clients like PowerPoint presentations. We're adaptable to that. Others like to see it in Excel formats, and both of our teams are well versed in providing the client with what they want so that they can have a better experience. And it also facilitates the movement of the project through both service lines, because you're often working in parallel, and so it just expedites things to be able to have better communication, and that facilitates a more successful project. It's on time and on budget, because the client's not like, well, wait, I got to work one way with this group and one way with another group. We've really done, again, a lot of work in trying to ensure that from the contract to the development data to the validation data all seem to be under one kind of KCAS process. And it's an important feature, and one it's still evolving, but it's definitely getting better and better every day. Yeah, fascinating stuff, John. Maybe we can continue to drill into the kind of the new entities, and one of the real kind of benefits of working with KCAS Bio is it's all under one location, right, John? I think that's a really important piece, in that it allows us to be able to, we often talk about a tool chest, and we having the proximity that we do. These are groups that can communicate. They're right across from each other. We really do try to remove any sort of silos. So we're able to do like feasibility studies real early to help determine what's the best platform or instrument to successfully measure we're kind of talking about PK specifically, but it also applies to biomarkers. And that's the power of having a hybrid mass spec team that works closely with a ligand binding assay team, and vice versa. So very timely, very cost effectively, we're able to start with one platform, or if you choose to invest starting with both, then it becomes a race. We like those. We're very competitive. It's kind of a fun little thing to do. But that becomes a real advantage, because I don't know. There are CROs that offer both, but they're often at different locations. And again, clients have told us it just feels like you're working with two different companies, and a little bit of a headache. Where we kind of avoid that.
I mean, again, it comes back to the fact that if we're talking that kind of, that, say, for example, an ADC or whatever, you have presence from the hybrid LCMS team and the LBA team, or scientific advisors, within that first call. And so you've got experts presenting, experts there to talk through the client's needs, but also what both technologies can offer, and not even why you may want to go in one direction or the other, but if there's issues, why one might be if there's issues using one, we can immediately pivot to the other, and just having that build that confidence. I see it because I like to join those calls as the small molecule person, but to hear experts from both LBA and hybrid talking how they could work through the large molecule piece, both talking knowledgeably and interacting non-competitively, it really helps build confidence with the customer very early on.
Yeah, but it gets, so whether you start with hybrid mass spec or LBA or vice versa. It gives you two very important things. One, you get to know the client's drug, right? How it's
Hmm.
behaving in a bioanalytical method, as well as the critical reagents. So there, even if one platform falls a little bit short, you still can glean information from that, so that you're not reinventing the wheel when you go from one platform to the other. And again, that's a time saver and
Sure.
a cost
Yep.
saver. So that's a
Yep.
really important feature. And John, maybe as we move through our agenda, it might be a good place to kind of go into some of those, just the LBA versus hybrid mass spec approach. And unless you had more to dive into about the entities and the location and
No,
so
no.
forth. But
I think that's absolutely.
yeah,
Take it wherever you want to go.
yeah, no, I think one of the we it seems to ebb and flow, but we've been getting asked more often which platform, right? Like
Hmm.
binding assays and hybrid, and there's this big push out in the field to say that the default should be hybrid LCMS for measuring total antibody and total ADC. But the problem I have with that, and there's this kind of biotransformation where there might be some nicking of the drug and so forth, but the capture step in hybrid is no different than the capture
Sure.
step
Exactly.
in LBA. So I think it's a little bit inflated, because I had someone on the call say, hey, the other, someone else had said, oh no, it should all definitely done by hybrid. But some of the when you really start to tease it apart, they talked about this, whatever group was pushing hybrid. They talked about creating a whole bunch of critical reagents to ensure the hybrid was working well in that capture step. Well, that's the same thing you can use in LBA. It
Yeah.
was
Yeah.
a fascinating. I was almost, I don't want to say confused. I was a little disappointed. In fact, I do understand that there are some times when hybrid is going to be the platform of choice, especially if there's like a lot of promiscuity with the reagents that can overcome some of that. But often doesn't matter if your drug has a lot of homology to something endogenous. It doesn't matter the platform. It still can be very problematic, and there are some ways you can try to deal with that, meaning taking a baseline sample and kind of subtracting that out, right? But it was just, I think there's a misnomer about how the advantages of hybrid over LBA. It's just that capture step is still the same challenges exist, and the same tools to resolve it are effective on both platforms. So
Sure.
it was just one of those things where, as maybe I'm a little biased towards LBA, obviously, but as somebody who really appreciates the power of hybrid mass spec, it just, I think it's maybe I'll summarize here. I think it's just a little bit right now. It's a little bit overblown how hybrid can solve things, because ultimately that capture step is still the same challenge. It doesn't matter if you're doing a bead-based capture on some sort of kingfisher or whatnot versus capturing inside of a plate on an LBA assay. It's still the same issues with trying to resolve that. And then maybe I'll let you jump in. And then there's a few other kind of misnomers that are out there around sensitivity, but I don't know if you wanted to comment on in general the capture step and all that.
I mean, I agree with the commonality of the capture step. I think where hybrid has more of a maybe more of a application is where you've got a shortage of other reagents for detection, etc. And then if there's other interferences on your LBA assay, and I don't know how often that happens, but at least by having the two side by side, you can easily pivot from one to the other, and so that to me is like. The longer I've been here, the more I've been open in terms of thinking, well, we can go either. I really don't mind which, so long as we get good data off that approach. I mean, I'm tend to be biased towards mass spec because I know what you can do with mass spec, but ultimately you're there to answer a question for a customer, and,
Yeah, sure.
and you want to select the appropriate technology that will give them the answers they need to move forward.
Yeah, and you're spot on that the mass spec doesn't need a second reagent for detection, but that concept that the detecting antibody is super challenging, or whatever sort of beliefs people might have about it. There's a lot of things you can do with prep of the sample that can remove some of those interference steps, like an acid dissociation could be
Sure.
a big part of that. And then also, as long as you can pull the protein out with a capture, that detection step's a little bit easier to optimize, because even if you're looking at a piece that, even if it is an FC, for example, in a total antibody assay, there's a wide range of different antibodies you can choose to ensure you're not seeing any sort of biotransformation, or even coming back with different anti-id could be a potential solution. And this theory biotransformation is, the body kind of nicks antibodies. I got news for you. If your body's nicking antibodies, your entire immune system could be compromised, if you know what I mean. It's just, antibodies are pretty hardy, is what I'm trying to say. They're kind of designed in such a way that, when in circulation, the binding region and even the FC region are somewhat, I don't know if protected is the right word, but there is a case for when you have good efficacy of the drug, you can create a microenvironment that is hostile. But again, if that's happening, it's still, the capture step is going to be impacted. So I'm not quite sure. I'm convinced that the affinity capture step isn't impacted any different than a ligand binding assay capture step. But it's a fascinating approach, and certainly something that we like to do early and often, John. That's the answer. Go ahead and look at it early to make sure you don't have any issues with one over the other. So great topic. I'm sure we could do a whole discussion on that, but
I'd probably draw Dawn in for that one too. That would be widen the discussion to a bigger team.
Yeah, no, maybe for the sake of time, let's move on to our next topic, which is really one that kind of to me has been quite a hot topic in the recent years, and that's microsampling, right?
Sure. Yep.
You've got a lot of experience there, but I'll let you start, and then I'll share my.
The reason why I throw that in, I mean, again, we had Neil Spooner on, last year. We talk about microsampling and patient-centric sampling, things like that, and it's still, for us as a bioanalytical lab, we're building experience with it, but it's still relatively uncommon in terms of number of requests. But because we're able to talk to each other, if someone comes to us with a request for, have you dealt with this sampling approach, both teams have experience, but it's obviously complementary. So there's a knowledge base to pull on. If someone comes to us with a new device, we're immediately going to ask, the LBA, have you worked on that, and what experience did you have? So like I say, it's by having the consistent communication, it's a pooled knowledge on these new patient sampling approaches, rather than, again, being isolated and having your own different operating as silos. You don't pool knowledge the same way. So again, it gives, having that pooled knowledge gives you it gives you warning of the kind of issues you might encounter, because you might not necessarily have worked with the technology directly in LCMS, but if someone in LBA has already used something similar, then we've got something. We've got a more elevated a starting point.
Yeah, no, we're very lucky to have experience in both LBA and LCMS with these microsamplings. And for me, I want to break it down by like a preclinical versus clinical, right? And in the preclinical space, there is a big push to reduce the number of animals.
Sure. Yep.
Recent sort of talk about trying to really move away from animals altogether, although I'm still a little hesitant. I think we're years away from that.
I agree.
But what we certainly can do is reduce the number of animals, and that's where microsampling becomes very powerful. And but when you take a sample at 20 microliter sample, and you generate maybe 10 microliters of plasma, that's still a very small amount to work with. But we're very fortunate to have some experience there, and I do think there's a large movement. There are some companies who are exclusively moving towards that in the preclinical setting, and it just allows for really not having to pool mice and sack them and things like that. It really can allow you to have just increases the quality of the method as well. And if you're using a single animal versus pool animal,
exactly,
as well as just the sheer number of animals and the humanity of it all is really a wonderful advancement in our space. But those sort of capillary microsamplings, John, right?
Yep, exactly. Yes.
And so that's great benefit for preclinical. And then when you go over to the clinical side, it's these TASOs and Mitras and other kind of microsampling devices that help with not having to go to the hospital. You don't have to stay in the clinic to kind of take a sample. You're able to do it from your home. These devices are pretty seamless to be able to use some of them without having to have a hospital visit. So all of that is really, as you mentioned, Neil, and that group's desire to kind of revolutionize how we're taking samples. And it was really powerful. I hope our audience goes back and listens to that episode. And I can't recall what number it is, but when Neil had said, "Hey, we haven't changed the way that we've taken samples for clinical trials in 50 years," it was like, whoa, that was really eye popping. So I don't know if you want to share your thoughts on that, but it is something where it's this microsampling is really going to be the forefront in the future for not just clinical, which is where the focus, in my opinion, had been, but it's really focusing on preclinical with the initiative to reduce the number of animals.
No, I agree with everything you said there. I mean, that ability to if you're taking small samples, to serially sample from animals to really cut down the numbers is great. So building that experience with those microcapillary tubes is there's obviously it's a different mindset to start doing it. Once you get in the groove, it's great to deal with for us as a bioanalytical group.
And you're just going to see more and more advancements in the sensitivity of the mass spec using less volume. That's going to be why mass specs are getting more and more sensitive. And again, I think on the LBA side, we've got a lot of experience microsampling for PK, and that's there's some ultra sensitive ways to do that. And maybe the new ELISA really holds a lot of benefit for that. And then even on doing ADA assays, we've had some success there, and that is. The way you treat those samples can be it's critical to try and mimic how the patient is, any sort of Tasso or Mitra device, how they're being shipped, how they're being stored. There's all sorts of things that are small nuances that we've been able to iron out in terms of even just making sure the sample comes to us a certain way. And then when you go to do the extraction off of it, there's a lot of little details that we've been able to work out to ensure we can get good sensitive methods on the LBA and the mass spec side. So great stuff, John. I think we're kind of winding down on breaking silos. And one of the last topic here, I think, is a real important one, and that's program management, right?
Yep, exactly.
This having a program management is a real key part of the communication process, and being able to kind of unify each of these service lines. And being a program manager is an unglorious position, I think, but I really do appreciate our team here. And it really does help with having teams that if the scientists aren't unified, then it makes it even more difficult on a program manager. But here at KCAS Bio, having teams that come to calls with clients, doing updates in a similar fashion helps streamline the process. But certainly a program manager is really key to your success I think at any company, not just ours, because they do have to kind of herd cats, right, John?
Exactly. Yes, it's you're right. It's a tough position, but to be there as that unifying voice is really, again, helpful to the customer, because again, what we do is all about customer service, and so that they're vital in terms of that presenting, collating and pulling together the different teams and ensuring that what we give to the customer is consistent. So really, really crucial to the whole success of the execution of our projects.
Well, what's nice about being a program manager here at KCAS Bio is you have a scientific advisor that you can turn to first, right? To help with responsiveness and really helping to answer questions with allowing our scientists and, operational leads to focus on providing good quality data, because a good percentage of the questions you and I and others on the scientific services team and scientific advisors can answer. But obviously, we don't, a program manager can reach out to a PI or anybody on the operational side to get a question answered. But the first step is to kind of come to us to help with that. Sure. And it really provides them with a lot of support. I think if you asked our program managers and those that have worked at other companies, or, my wife's a PM for a CRO, that she doesn't have the resources that our program managers have here. And she at times is like, man, that your PMs are real lucky to have the number of resources they have. Because sometimes if the operations is focused on something else, it could be potentially hard to get a hold of them, where at least you have a scientific advisor that can either help translate some of that, right? Sometimes the questions
Yep.
can be answered by us, and other times we're able to take those questions and interpret them a little better, and that allows the PM to really quickly ask them more concise and clearer. And again, that
Sure.
just gets back to
Exactly.
better communication, better product.
Yep.
Well, great, John. I
Okay.
don't
I
have
think
anything else. Yeah.
no, I don't have anything more to say. I'm glad we had a good chance to talk about just the philosophy at KCAS in terms of consistency. But I think we can move on to closing out for this podcast.
Yeah, John. Why don't you tell me what you got coming up this weekend or in the coming weeks that are of interest?
This weekend will be relatively quiet since I only just got back from Europe on Monday. So yeah, jet lag hasn't been too bad this time, I'll be honest. But yeah, I'm still sort of basking in the glow of a really pleasant 2 weeks in England and France. Yeah, next weekend though is my wife's birthday on the Saturday, so that will be much more of a much more fun going on there. We don't actually know what we're doing, but at some point we'll obviously figure out something for the day, and the kids will be around, so we'll do something good then. But this weekend I think will be relatively quiet, and we're still waiting for it to warm up. There was ice on my car this morning.
Oh wow!
Yeah, I know. It's like it's so we because basically it was nice yesterday, and then started raining yesterday evening, and then I went out this morning, and my car had ice all over it from the frozen rain, and then at some point I need to get my car fixed. So probably one of the things I'll have to do is I have to take some pictures to get a collision damage estimate, because when driving home on Monday, I was driving down a long straight of road, and I was, as usual, wasn't really hanging about, and there was a squirrel in the middle of the road, so I tried to avoid the squirrel, which it did that whole where they zigzag and do something stupid, and in doing its avoidance, it somehow jumped into the grill of my car, so it's taken out my car has like 3 front grills. It took out a chunk of the grill, took out 2 motion sensors, and so the car is messed. Well, basically, there's the broken grill, but the car is now messed up because every time I reverse it, the front motion sensors start screaming because they can't quite figure out what's going on. So I really need to get the estimate to fix that, so I can get it fixed and get the car sorted again. But that will probably happen. I'll take pictures of that this weekend to hopefully get it fixed pretty soon, because I can't live with that.
It's a little bit of a boring weekend for both of us. I think I'll probably watch some basketball. I know I got some friends that are Iowa fans, and Iowa made it to the Elite Eight, so there'll be already planning for a fun party on Saturday. So I don't really have anything exciting other than in about 10 days, I'll be heading to Las Vegas for the Frozen 4. I went last year.
Okay, nice.
Yeah, my brother and my nephew, it'll be their 8th year. And this year, my nephew actually got picked up by the college network television, and he'll be broadcasting some games. And it seems like I think he went to something like, I don't know, I think he went to more college hockey games than anyone in the U.S. And
wow.
So they kind of caught him, and he's really, he knows it well. He loves it. He's got a lot of passion. It's easier to be a college hockey fan when you live in Massachusetts. You can,
sure,
a 2-hour radio, 2-hour drive, you can go to a lot of games.
Yep.
And so he's going to be kind of broadcasting live from there, and should be fun. That's going to give us some cool access. And of course, it's Las Vegas, and it'd be great to hang out with my brother. And I think there's about 6 of us going. So some other friends. And then I think my nephew has some colleagues or friends of his that were from Massachusetts that live out there. So that's a real exciting thing for me. But this weekend's pretty yes. Just kind of quiet.
It's funny that being in Europe, I completely missed it. It was the NCAA tournament, and I didn't realize it until yesterday. Something popped up on my phone talking about the Sweet 16. I'm like, Sweet 16, really? It's now? Yeah. Oh yeah. So I have not seen anything about it at all. So I
No, those are.
couldn't tell you who's in the Sweet 16 or the Elite Eight, or who is through it. Apart from now, I know Iowa are there. I couldn't tell you any of the others because but like say, being in Europe, that just wasn't a factor, you know.
Yeah, Iowa, Illinois, Arizona, those all advanced yesterday, and the fourth one was, I think, I can't recall who the fourth one is, but there's obviously the remaining Sweet 16 games tonight,
Sure,
and then the
yeah.
Elite Eight will be over the weekend. But really, it's a fun time of year,
Yep,
and
yep. Oh,
it
absolutely.
doesn't surprise me the Europeans aren't into it. But I think I feel like it's a lot outside of the Super Bowl. It's the most lost wages in here in the U.S. is due to the NCAA tournament. But John, you mentioned the weather. We had like close to 90 degrees yesterday. We've had a couple record highs over the last week in terms of weather. So I think we dropped down a little bit today. It's chilly. It got into like maybe the 40s, low 40s. But it's going to warm back up again. And we're like green grass and things are budding. It feels like almost spring already. Well, I guess it is spring, right? Spring has sprung.
Yep.
The golf course looks good. And with a bit of luck, maybe I can get out and play around. But John, been a wonderful podcast, and I don't have anything else to add other than I'll see you in the funny pages.
Well, we'll see you in podcast number 100 in the next month or so, and that will be. I'm sure we're going to do something special, but not sure what that will be. But
stay tuned.
That'll be quite a milestone to hit.
Yeah, no, that's something we should be very proud of, is our 100th podcast, and that is very exciting. So stay tuned.